Hey everyone, welcome to lesson 15. So in lesson 14, we went over innate immunity, and now we're going to move into adaptive immunity. So when we think about the two branches of the immune system, innate immunity is the basic defenses. Think of these as things like moats and walls. And then when we move over to adaptive immunity, that would be something like archers and soldiers that can move around and tackle different issues as they come up. So we discussed innate immunity being things like macrophages, dendritic cells, granulocytes. Those are the ones that generally cause allergies, and different ways that they essentially search through the system, gobble things up, and then present it to the adaptive immune system. So I talked about this presentation process, and we're going to jump in with discussing exactly what they're presenting. So we need to understand a term, and that term is an antigen. So antigen. Not like a person's name, but GEN. So that is just the entire word meaning something that is foreign or non-self. Usually these are proteins, and if it's not a protein, it could be a polysaccharide like a sugar. Remember that a lot of bacteria have polysaccharides on them, and that can also be recognized by the adaptive immune system. Each antigen, if you think about an antigen as a full protein, has many, 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 many little regions on it. You know, you think about a huge protein, the biggest protein in the world. It is this gargantuan thing that's many, many kilodaltons, larger than something like a ribosome subunit, right? And those are already large proteins. And then you have all of your enzymes and everything else floating around. So those proteins are made up of many, many amino acids. Well, those short bits of amino acids, around 8 amino acids long, are what we call an epitope. So an antigen is the whole protein or the whole sugar or everything else. An epitope is a specific sub-region of that the body is going to recognize. And the reason we hone in on epitopes is because, much like during COVID-19, the overall protein may remain the same, but those little epitopes might change. And we're always looking for epitopes that we can signal into, identify, attack with the adaptive immune system. Now, hopefully that makes sense. So antigens are whole protein. We'll just keep using proteins as the example because it's the easiest, I think, to understand. And the epitopes are little segments of that protein. Well, how do we recognize those? The main way we're going to recognize those and build defenses against these epitopes that are on the antigens is with antibodies. Typically, you're going to think of antibodies as these Y-shaped proteins that have these two little heads that attach to epitopes. And that is what we call an IgG or a immunoglobulin G subtype. This is usually what is shown because it's the most abundant in the human body. It is responsible for long-lasting immunity. It is what we would give someone if they were suffering from some sort of medical condition. One thing that's typically used is we have IgGs or immunoglobulins against cancer. We also have these against viruses. So these are treatments that we use, and those are generally going to be based on these IgG or these immunoglobulin subtype Gs. Now, there are other subtypes as well in these different antibody subsets. So, IgGs are the most abundant. They circulate in your blood, and interestingly, they can cross into through the placental barrier. So, they can go through the placental barrier and actually protect a fetus. Then we have IgAs. These are found in your tears, your saliva, and also in breast milk. So, they're going to be your kind of first line of defense. And they're essentially this Y-IgG clipped to another one. So they clip at the tail. So you have this Y and then you have this other Y and it clips together. and it makes this IgA. The reason it does that is because this allows it to have more sensory ability to bind, because instead of having two heads, it now has four. And that's important somewhere like your mouth, where you have a lot of bacterial turnover. You might be eating food, things like that. Then there are IgMs. These are actually going to be the first antibodies that your body produces to a new infection. And these, instead of being dimers, so instead of having two, are actually pentamers. So they have five IgGs all connected together. This allows them to come into contact with many, many more antigens and in turn epitopes than a simple IgG would. So we can think about this as you have your IgM first, which is five put together, five antibodies put together, and that's used to kind of sense what's going on in the region. And these will have a lot of diversity. They'll hit a lot of different epitopes on different antigens, and then it gets funneled down to either an IgA or an IgG. And these are more specialized. We've gone through the process of secreting the IgMs, those pentamers, and sort of surveilling and seeing which things, when we block them, actually make us feel better. And there's a lot of mechanisms behind how this works, but that's the basics of it, is that you produce the IgMs first, those pentamers, they detect what's there, they see what can be blocked, and then after that you get these IgAs that are in your saliva and other secretions, and then IgGs which circulate in your blood. Now, of course, nothing can ever be that simple in biology, so there's also two other antibody subtypes that you might run into, but know that IgM, IgAs, and IgGs, especially IgGs, are the ones you're going to want to be the most confident about explaining for this lesson. So, IgEs are, or immunoglobulin type E, is involved in allergies and defending against parasites. Notice again, we have that crossover, like with the granulocytes, that if it defends against parasites, it also seems to cause allergies. And there's a lot of thoughts around why this happens, but the most likely is we live in a very clean society. Now, you might think of a lot of things as dirty, but we're not walking around with bare feet in mud. We're not, eating things that are just completely unwashed or undercooked. So our exposure to parasites is extremely low. I would be very surprised if anyone that you know has had a parasite unless they traveled outside of the United States. And the reason for that is because everything is sanitized in what I think is a good way, where we don't have to deal with parasites a lot, but we also still have these IgEs that circulate and these granulocytes that circulate, and they're looking for something to do. And the hypothesis is, because they're always there, they do find things that look like parasites, and then they react. And that's where allergies come from. There's also IgDs, so D as in dog, IgDs, and these act as receptors on immature B cells. So when we're looking at the flow of how these antibodies are made, you actually have the IgD first. So all antibodies are made by B cells. In the adaptive immune system, there's B cells and T cells, and antibodies are produced by B cells. On your B cell, you have these IgDs. So they're like IgGs, and they just sit on the surface of the B cell, and then it contacts something, and that B cell says, there's something here. I need to start pumping out different antibodies. And that is typically going to start out as IgMs, and then later will turn into and differentiate until they produce IgAs or IgGs. Very complex process of how all of that works, but antibodies are our main adaptive defense system against viruses, against toxins. So actually, they are used to neutralize toxins. They are used to mark things for what we call opsonization, that eating or phagocytosis by things like dendritic cells or macrophages. So the innate and adaptive immune system is always talking to each other. It starts with the innate, and then the adaptive turns on, and then it becomes a cyclical process where the adaptive immune system says, hey, I'm marking this thing with antibodies. I want you to get rid of it. I want you to eat it and digest it and spit all of its parts to tell the immune system, hey, it's still here. We still need to be active because, they can't actually coordinate in any logical, thoughtful way. So they have to have all these signals for what they do. And this gets us into antigen processing and presentation. So your different lymphocytes, these white blood cells, or these predominantly T cells, are going to have antigens shown to them. They need some sort of presentation to say, hey, this is something we should be thinking about, worrying about, and maybe reacting to. That typically is going to be through macrophages and dendritic cells eating something, taking all the little pieces and parts, going to a lymph node. So you might know that those get all swollen when you're sick. And the reason for that is you have all this trafficking that goes into them, and there is an influx of cells that gunks it up where they're all trying to talk to each other, trying somewhat like a busy DMV on the weekend, right? So everything's in there trying to communicate. And all of these antigen presenting cells, so those macrophages and dendritic cells, are in there saying, hey, I found something. I found something. I need you to look at this. And there's two different ways that these can be recognized. One is by MHC1, and this is on all what we call nucleated cells. So this presents antigen from the inside of a cell, such as a viral protein, to a cytotoxic T cell. When this MHC1 reaction happens, it's essentially a cell saying, I am so sick, I'm so infected. Please look at this thing, and if it is bad, kill me. And then a cytotoxic T cell comes and then does the killing. MHC2 is how we get this robust immune response, though. This is when antigen presenting cells like those dendritic cells and macrophages, and even B cells, because remember, the B cells have that IgD and they still need to talk to T cells, all of those take and present the antigen to what we call helper T cells. These are the T cells that decide we are going to react to this or we are not going to react to this. They're called helper T cells, but they are in charge. They're in charge of the whole immune, at least the adaptive immune process of saying, hey, this is what we're going to respond to. No, we're not going to respond to that. So when the presentation happens from the dendritic cell, from the macrophage, from the B cell, it's to a helper T cell or a CD4 positive T cell. And then that T cell says, hey, this is something that we need to react to. So we're going to start reacting to it, right? And this process is being done with majority naive cells. And we call them naive because they don't recognize anything in particular and they're not matured. Um, and this gets into a lot of detail. But essentially, the path for your adaptive cells starts in your bone marrow. So that's the tissue in your bones. It produces your B cells and your T cells. The B cells actually mature in the bone marrow and then get released. They're still what we call naive because they haven't been fully activated yet, but they will distribute into the bloodstream, start circulating, and see, hey, do I recognize this? Do I recognize this? Is this something I need to recognize? Is this something I shouldn't worry about? And then your T cells actually go up to your throat in your thymus, and that's why they're called T cells. B cells are called B cells because they are found technically in the bursa fabricus of chickens, but that is the bone marrow of the chicken, kind of, sort of. But it works well because it's in our bone marrow and they're still called B cells. So you can think B cells, bone marrow. And then T cells mature in the thymus. So T cells come from the thymus. They're thymus cells. And then they go out and they circulate as these still naive lymphocytes until they're activated by some sort of antigen exposure. Now, this, of course, is getting into a lot of complexities of immunology, which is astoundingly complex. There's a ton of subtypes of these cells. There are multiple different steps to how these cells mature, and a class about immunology is an upper division class that I'll be honest, myself and other people I know who are immunologist trained will still struggle with some of these concepts. We have to go back to a book and double check and look it up because there's so much information. But there's a couple more things I want you to understand, and that's the type of T cells. Now, the T cells have three main types, but it's going to be helper T cells that are doing the orchestration of your immune response. These T cells are the ones that are talking to your antigen presenting cells, again, deciding which antigens are good and can just be left alone and we don't respond to, which is important because you have a lot of proteins in your body, a lot of antigens in your body, a lot of epitopes in your body. So all of those, we need some way to say, please don't attack these. And a lot of autoimmune diseases come from the helper T cell, or for some reason that wasn't contacted before this immune response was triggered, saying the wrong thing, saying you are good to attack this thing, and it turns out that's your connective tissue in your joints or something like that. So that would be rheumatoid arthritis, as you get all of this uh, attacking of immune cells into your joints because your helper T cells messed up. But they're super important because generally they're going to keep your body in balance and then attack only outside viruses, toxins, things like that. Then there's cytotoxic T cells. We talked about them a little bit. These are your CD8 positive cells, and they destroy infected and abnormal cells, whether it's infected with a bacteria or a virus or a protozoa or it's a cancerous cell. The cytotoxic T cells are the ones that are going in and actually doing that direct cell to cell killing, where you get rid of that infected cell. Then there are memory T cells. These are discussed a lot less generally in immunology because they just kind of happen in the background and no one really targets them for therapies. But these memory T cells provide a long-term immunity. They are helper T cells and cytotoxic T cells that recognize what antigens have been there before, what they need to respond to. So they're very, very important. Similarly, B cells can fall into this either you're getting activated with something new or you're getting activated because you recognize something old that you have already dealt with before. So T cell dependent activation is when a B cell says, hey, there is something that I think is bad. What do you think, helper T cell? And helper T cell says, yeah, that is bad. Make a bunch of what we call plasma cells and memory B cells. The plasma cells are the ones that are going to be short-lived and release a ton of antibody, and that's only their job. The memory B cells are going to say, I'm going to catalog this epitope on this antigen, and if I ever see it again, I'm going to activate, and I'm not going to ask you any questions, helper T cell, because we already know what's up. And that's how you get T cell independent activation of these B cells, releasing antibodies. Um, and that is generally a good thing, right? It's a weaker response, and you get all these memory associated antibodies that will circulate because it's recognizing something. And then when it does get a signal from a helper T cell saying, hey, you're doing the right thing, you need to really boost this up, then you get this very robust response. And that's why things like vaccines are so important. Getting the chickenpox vaccine or your COVID vaccine or your flu vaccine, whatever it is, it allows your body to start fighting before really getting the okay. And this okay takes anywhere between 7 days to about 2 weeks when you're first infected. Remember the curves where you get infected and then you actually feel sick, and that often is about 7 days later. And the reason you start feeling really sick isn't just because you're infected, but it's because your body's responding. It's inflamed, it's angry. So you get this 7-ish day infection curve. But if you already have responded, that 7 days is shortened to around 3 days when you get a robust response to something. And you might, feel sick really quick, but it's already killed what it needs to kill, gotten rid of the infection. So you get, 1 day of sickness instead of this long curve up and this long curve down where you're out for a week from something like the flu. And that's why T cell independent activation is so important. And this gets us into what we call primary responses and secondary responses. Your primary response is your first exposure, and this is days to weeks. Mostly you're producing IgM that then turns into IgG and IgA over time. You're not having robust responses. It's not fast. Your secondary response is faster and stronger because of these memory B cells and memory T cells. And then your response is dominated by this IgG response because it knows exactly what epitopes to hit. It knows to hit them hard. It knows to hit them fast. And again, this is why vaccines are so very important because this allows us to get over sicknesses, even if we still feel sick. A lot of people say, I got the flu vaccine and I still felt sick for a couple days. And the answer is, yeah, you did feel sick for a couple days from the flu. But the reason you felt sick for a couple days is because you had a really hard and fast response and you weren't knocked out for a week. Um, when we talk about adaptive immunity and vaccines, we really have to break up what our immune response is based off of and how we categorize these different responses to different things. Um, and there's four different kinds of overall immune responses we're going to talk about. The first is naturally acquired active immune responses. This is from when you get infected. And this is really important because we need to know if the infection happened, you fully recovered, and now you have immunity to that infection. So this is a naturally acquired active immune response that you're having, and you get memory from that. The other type of naturally acquired immune response from the adaptive immune system is actually passive. And this comes simply from antibodies. The majority of this is going to be a mother providing breast milk to a child that has those IgAs in it, and that gives the child a passive immunity to everything the mother is generally immune to. But we can also think about this naturally acquired passive immunity from things like injections of immunoglobulins from other people. So if you have someone, for instance, that has COVID-19, they might get an injection if they're really, really sick, especially the early days of that. Or if you have a cancer, you might get a immunotherapy, but that's naturally acquired passive responses. You aren't making that yourself. So active means you're making it yourself, and then passive means that you are essentially it is being made for you. And this actually splits. So right here we have artificially acquired passive immunity. And this is when we get to that injection of antibodies or anti-serum. So, say you get bit by a snake, you will go get a anti-serum for the snake venom, and that's just a bunch of antibodies. It actually used to come from horses, and I think it still does come from horses, where you take the venom from the snake, inject it into the horse, and the horse is huge, so it says, "This is kind of making me feel bad, but I'm not dying like a human would." So I'm just going to make a bunch of antibodies against these toxins, get rid of it, and then we take that horse antibody and then we give it to someone who was bit by a snake to save their life. And that's an artificial way of doing it. Even though it has natural processes, of course, it's artificial and it becomes more artificial when we're designing something in a lab. And that's just a way to differentiate between naturally acquired, which is usually just mother to child when we talk about that, versus artificially acquired, which is the injection of an antibody from an outside source. Then there's artificially acquired active immunity, and this gets into, again, vaccines. You'll notice that vaccines are a big topic, and that's because they're a really good demonstration about how we can use the adaptive immune system to our benefit. So, artificially acquired active immunity comes from things like vaccination. And speaking of vaccines, we're very briefly going to go over the different types, and I'm going to kind of discuss why these different types exist and why we've moved away from some and moved towards others. So the FDA, I forget at what point, decided that they were going to prioritize safety over efficacy. And the reason for this is there would be clinical trials run, and someone might become severely debilitated or even die, and that was argued by the drug manufacturers, the researchers making this, well, yeah, they died, but it's a very deadly disease. So instead of 10 out of every 100 dying, it's 1 out of every 100 dying, and that should be acceptable. And for a long time it was. But as science improved, especially as all these biologics and antibody-based drugs started being made, they said, we have to move away from this. We have to get into, we need something that's very, very safe because once you start giving those drugs to tens of thousands, millions of people, that death that 1 in 100 becomes unacceptable. So they move towards safety above everything else. So originally we have what we call live attenuated vaccines. These were weak but living microbes that would replicate in your body. They would replicate some, but they're weakened. So anyone who is very healthy can fight them off, usually kids and adults, but the elderly, you would give these vaccines to them, and some people would at least, if not die, have very, very severe complications. So then we move to what we call inactivated vaccines. These are still whole microbes that were killed. They are safer, but they have the negative of it's a weaker response than these live attenuated vaccines. And the reason it's weaker is because you don't have that replication cycle that your body knows means something's wrong if you're getting hijacked replication by these different microbes. So the inactivated vaccines, safer, but a weaker response. And then we get into what is the modern day majority of modern day vaccines, and that is subunit slash recombinant vaccines. These are little pieces of an antigen, so getting to a handful of epitopes, and those are injected. They are extremely safe, but oftentimes they require 2 to 3 shots instead of just one, like the live attenuated or inactivated vaccines. So, see, we're giving this plus and minus, and the plus is going to safety, and the minus is going to efficacy. Not to say they aren't working, it's just you might need 2 shots or 3 shots instead of just one shot. Then we go to toxoid vaccines, and it's a similar idea as subunit and recombinant vaccines. Toxoid vaccines are toxins that you give to someone. They can't replicate. There's not a microbe making a ton of these toxins. They are just there, and your body disposes of them naturally. But of course, it's a toxin. So someone who isn't having a good response might have complications from that. And then we get into modern day vectored vaccines or mRNA vaccines. These instruct a cell in your body to make an antigen. And this gets back to that live attenuated step of replication, of having these proteins made inside your body. So it is replicating these subunits, these fragments of antigen, and those fragments are being recognized. And this is a really good way to do a vaccine. It is, however, unpredictable to a certain extent because I might have really good metabolism and make a ton of these proteins because I'm active and young and the mRNA gets into cells, usually muscle cells is how they're administered, that I'm working out all the time, big and strong. So I'm producing a ton of these antigens and I get sick. I feel terrible taking an mRNA vaccine. And that's actually true. Usually the next 2 days after I get an mRNA vaccine, I don't feel great. I do feel sick because of the immune response happening. So then we go back to subunit vaccines, which are again very safe and very effective, but it might require 2 shots instead of one. So we end up in this place where it's really up to the person getting the shot. Do I want to get 2 shots or maybe get 2 days of feeling not great from an mRNA vaccine? But at the end of the day, they're both very safe and they both work really well. So just to kind of summarize everything, because it is a lot, really focus on understanding what is an antigen, and remember that epitope is a small part of an antigen. And an antigen can be usually a protein, but it can be other things like polysaccharides, like sugars. Next, know that antibodies are the main way that our adaptive immune system is going to have a robust long-term response and going to not just respond initially, but going to respond with long-term memory. It's going to be these antibodies against specific epitopes that clear out viruses, that clear out bacteria, that clear out toxins from our bodies. Understand that you have to present the antigens before you can get to that process, and you have to have mature T cells and B cells to do so. Know that B cell activation has two different arms, T cell dependent, T cell independent, and know that the helper T cell is the one that's orchestrating this entire immune response, at least when we're talking about it right now. It gets way more complicated. And then try and remember the different types of vaccines and why some have gone out of vogue, so to speak, and why others are used and pursued in modern day. I will say inactivated vaccines, some still exist because the cost of taking all of those proteins in a microbe, producing them, and then giving them to someone is way too high. And there's other things in the inactivated microbe that actually make it so you respond better to that vaccine. So it's not like these aren't always used. Sometimes they still are. Sometimes even new vaccines will use these old methods because it's what works best. But know that the FDA is focused on safety, and that's why most new vaccines are subunit vaccines or these mRNA vaccines. So that is lesson 15. We have one more lesson after this that we're going to kind of connect everything from the class together. But I think immunology is absolutely amazing. If you have any questions about it, the complexities, please let me know. And until next week, I hope you have a fantastic time with this lesson. Bye.